Welcome to today's workshop, Teaching Enzymology with the Protein Data Bank from Pandemic to Paxlovid. This workshop is hosted by the American Society for Biochemistry and Molecular Biology and has been facilitated and organized by the RCSB Protein Data Bank. My name is Kirsten Block, and I'm the Director of Education, Professional Development, and Outreach for the ASBMB. Before I turn over the workshop to our facilitators today, I'd just like to mention a couple of housekeeping notes. So this workshop is structured as a Zoom meeting rather than a Zoom webinar, and that means all attendees do have the ability to turn on their microphones and their cameras as needed. However, we do ask that you please be respectful of our facilitators and other attendees and keep yourselves muted unless called upon at various points throughout the session. And we welcome and strongly encourage that you make use of the chat feature to engage with the speakers as well as with other attendees. You will also see some polling throughout the session, so we encourage you to participate in that manner as well. So with that, I happily will turn this workshop over to our facilitators today, starting with Dr. Stephen Burley.
Thank you very much, Kirsten, and my thanks to the ASBMB for co-sponsoring this webinar with the RCSB Protein Data Bank, which I lead. Today's topic, as Kirsten mentioned, is teaching enzymology with the Protein Data Bank from pandemic to Paxlovid. So today's agenda is provided on this slide. I'll introduce the Protein Data Bank and SARS-CoV-2, which regrettably doesn't actually require very much of an introduction. And then our colleague and longtime advisor and more recently very intensive collaborator, Professor Paul Craig from the Rochester Institute of Technology, will talk about exploring SARS-CoV-2 main protease using the rcsb.org web portal. And then a member of the RCSB Protein Data Bank staff, Professor Shuchismita Dutta, will talk about making connections using rcsb.org. We've structured the time that we have with you in such a way that there should be plenty of opportunity for discussion thereafter. So my plan is to tell you where the Protein Data Bank, the PDB, has been, what we've accomplished over more than 5 decades, and the critical role that structural biologists and the PDB together played in fighting the COVID-19 pandemic. Now, the punchline of today's entire event was put quite succinctly by Dr. Anthony Fauci, former head of the National Institute of Allergy and Infectious Disease. In the New York Times Magazine earlier this year, he cited the importance of 3D biostructure information and said, "Show me a person who's vaccinated, got infected, took Paxlovid, and died. I can't find anybody." I'll now explain why Fauci made these statements and put the global efforts of structural biologists and the PDB into context. The Protein Data Bank was established as the first open access digital data resource in all of biology. It was founded in 1971 with just 7 X-ray crystal structures of proteins, and it's been continuously funded by the United States government ever since. 4 of the early structures motivating protein crystallographers to establish the PDB are illustrated in the right panel. Since its inception, the archive has grown more than 30,000-fold to become the single global resource for experimentally determined atomic-level 3D biostructure information. At present, the PDB houses more than 210,000 structures of proteins, nucleic acids, viruses, and macromolecular machines. Recognizing the global importance of the archive, it has been managed jointly since 2003 by the Worldwide Protein Data Bank Partnership, including data centers in the US, Europe, and Asia, and 2 specialist data repositories for 3D electron microscopy and NMR spectroscopy, respectively. Once the data have been archived in the PDB, our rcsb.org research-focused web portal serves as a one-stop shop for open access to more than 210,000 experimentally determined PDB structures, alongside more than 1 million computed structure models coming from AlphaFold DB and the Model Archive. We deliver public domain 3D biostructure information to many millions of basic and applied researchers, educators, and students around the world at no cost and with no limitations on usage. For the avoidance of doubt, the provenance and the reliability of both types of 3D biostructure data, experimentally determined and predicted, are clearly identified on our website. This slide shows the rcsb.org homepage that you'll become familiar with during the course of this webinar. Highlighted within the red ellipse is the toggle that allows you to opt in to access the more than 1 million computed structure models, or CSMs. Moving the toggle from left to right enables combined PDB and computed structure model searches from the top bar, which is present on every web page, and when using our powerful advanced search option, which supports full Boolean logic. PDB data are essential for responding to emerging viral pathogens. Since the early 2000s, the world has faced down 3 major outbreaks of coronaviral infections that jumped the species barrier to humans. First detected in 2002, the SARS coronavirus infected about 8,500 individuals worldwide, killing just over 800 people between 2002 and 2004. The second coronavirus epidemic, caused by the Middle East Respiratory Syndrome, or MERS coronavirus, struck in 2012. To date, it has killed more than 900 individuals and remains a public health threat in the Middle East and parts of Asia, where it is endemic in camel populations. Despite these 2 warning signs, very few countries actually prepared for the possibility of a much more serious coronavirus epidemic. Fortunately for the global community, structural biologists and the PDB laid the groundwork for a successful response to the inevitable third coronavirus wave. I say inevitable because after MERS hit, the experts knew it was just a matter of time before yet another coronavirus jumped the species barrier to human. For SARS-CoV-2, the third coronavirus, effective mRNA vaccines were designed and antiviral agents were discovered with the benefit of open access to PDB structures of SARS-CoV, MERS-CoV, and SARS-CoV-2 proteins. SARS-CoV-2, the unseen enemy, is shown on the right within a respiratory droplet in a haunting painting by our artist in residence, Professor David Goodsell, who has authored the RCSB PDB Molecule of the Month series for more than 20 years. Since the very first SARS-CoV-2 protein structures were deposited to the PDB in 2020, the RCSB Protein Data Bank has been hard at work responding to the ongoing pandemic. Together with our worldwide Protein Data Bank partners, we prioritize complete deposition, rigorous validation, and expert biocuration of COVID-19 related protein structures, and we strongly encouraged immediate pre-publication release of these data. I'm pleased to report that most structural biologists did release their PDB structures to the public before they'd actually been published in scientific journals. We created a COVID-19 coronavirus resources page on our rcsb.org web portal, which is updated weekly with each release cycle that adds approximately 300 new structures to the archive every Wednesday at 0000 Universal Time Coordinated, which is at about 8 p.m. right now on the East Coast of the US. We also developed education and outreach resources, such as this hand-washing animated video, viewed more than 500,000 times, and a series of RCSB PDB Molecule of the Month articles co-authored by David Goodsell, featuring SARS-CoV-2 proteases, the RNA-dependent RNA polymerase, and the viral spike proteins. The organization of the SARS-CoV-2 genome is depicted schematically in this slide. All coronavirus genomes are very long, single-stranded, positive sense, 5' capped, 3' polyadenylated messenger RNAs that are ready for translation by host cell ribosomes. Most of the non-structural proteins are actually expressed within a pair of related polyproteins. Individual non-structural proteins are excised by 2 SARS-CoV-2 proteases. Both are non-structural proteins themselves and are part of the polyprotein. The papain-like proteinase cuts at 3 sites in the polyprotein, denoted with the dark blue down arrows in this image. The even more important main protease cuts at 10 sites denoted with light blue inverted triangles that you can see here and through here. The right panel of this slide summarizes what is now our nearly complete atomic level 3D knowledge of SARS-CoV-2 protein structures that is embodied in more than 3,000 PDB depositions that were received and made public since early 2020. The left panel shows an artist's rendition, again from David Goodsell, of the lipid bilayer of the SARS-CoV-2 virus fusing with the plasma membrane of a soon-to-be infected cell in this interestingly colored painting. This viral entry process is blocked by the immune response to mRNA vaccines against SARS-CoV-2 that I'm going to describe in a moment. The very first PDB structure of a SARS-CoV-2 spike protein, and that has ID 6 Victor Sierra Bravo using the NATO alphabet, was released to the public in February 2020, about a month after the sequence of the viral genome became public. Today, there are nearly 1,600 spike protein-related structures in the archive for SARS-CoV-2. Like the earlier coronavirus spike protein structures in the PDB, they provide important insights into receptor binding, fusion of the viral lipid bilayer with the plasma membrane, and vaccine and antibody design. The very first mRNA vaccine designs generated in 2020 were actually based on PDB structures of the SARS-CoV and the MERS-CoV spike proteins, which are very similar in amino acid sequence and 3D structure to their SARS-CoV-2 counterpart. When designing Moderna's inaugural mRNA-1273 vaccine encoding the SARS-CoV-2 spike protein, the researchers at NIAID and the company reported relying on PDB structures of double proline mutant forms of the SARS-CoV and MERS-CoV spike proteins. These mutations stabilize the spike proteins in very highly immunogenic pre-fusion conformations. I believe that the vaccine designers at BioNTech used the same information because their inaugural mRNA vaccine also encoded a SARS-CoV-2 spike protein bearing the identical double proline mutation. You've got to love the PDB. The rest of the story is well known to all of you. The two essentially identical, highly effective mRNA vaccines against COVID-19 were brought to the market in record time by Operation Warp Speed. To date, these vaccines and their successors have been administered to more than 5.5 billion individuals worldwide with impressive results. The vaccines designed with the benefit of open access to PDB structures are credited with saving tens of millions of lives globally and preventing severe forms of infection in hundreds of millions, if not more than 1 billion individuals worldwide. You've got to love the PDB. As a physician, I urge you to consider being revaccinated with one of the updated mRNA vaccines that are now available. Revaccination will provide you with renewed protection as we move into the Northern Hemisphere winter. You should consult your physician to determine whether or not you're a candidate for vaccination. And you need to be aware that even now, between 50 and 100 people are dying every day in the United States from COVID, and the situation is likely to worsen as the weather gets colder and people spend more time indoors. Today, most of the fatalities are occurring in the unvaccinated elderly and in vaccinated individuals with various comorbidities who are at higher risk for serious illness and fail to secure a prompt antiviral treatment with either Paxlovid or molnupiravir. I'll move on to Paxlovid now. Paxlovid is the product of structure-guided drug discovery. The PDB currently houses more than 750 crystal structures of the SARS-CoV-2 main protease, the first of which was PDB-ID 6 Lima uniform 7, that was deposited to the archive by structural biologists based in Shanghai. This protein is the target of Pfizer's highly effective, orally bioavailable small molecule drug known as Paxlovid, which is actually a fixed-dose combination of nirmatrelvir, the active ingredient that blocks the action of the SARS-CoV-2 main protease, and ritonavir. The importance of ritonavir, excuse me, I should have moved to the next slide. The importance of ritonavir is that it reduces the likelihood of degradation of nirmatrelvir as the drug passes through the liver. Paxlovid has been approved for outpatient treatment of individuals infected with SARS-CoV-2. Ideally, it should be given within the first 5 days of the infection when it can be most effective. Now, it is the case that some individuals require not 1 but 2 5-day courses of Paxlovid to clear the virus from the system, but that should not be seen as a failure. It's just simply a question of dosing strategy in individuals with different viral loads. The clinical trial that was done by Pfizer to bring Paxlovid to the market used a 5-day treatment plan. And so we're committed as physicians to prescribing the drug initially for 5 days and then making it available again for another 5 days for individuals who do not completely clear the virus after the first course of treatment. So I'm going to turn the rest of the presentation over to Professors Craig and Dutta, who are, as I said, going to first introduce the SARS-CoV-2 main protease in much more detail using the tools that are available on rcsb.org, our research-focused web portal. And then Dr. Dutta is going to show you how to make connections using the rcsb.org tools. As I said at the outset, there'll be plenty of time for discussion at the RCSB Protein Data Bank. I'm sure the same is true at ASBMB. We believe that all feedback is good, and we welcome your feedback both during this session and in the exit survey that you'll be provided access to by ASBMB at the end of the event. So at this point, I'm going to stop sharing, and I'm going to go off camera and turn the floor over to Professor Paul Craig. Craig, Paul, it's all yours.
Thanks very much, Stephen. And I'd like to add my welcome to everybody who's joining us today. It was great looking at the list of participants and recognizing some of familiar names and lots of names I haven't seen before. So this is really great. The goal of this part of the presentation is to introduce or maybe reinforce the use of a number of RCSB PDB resources for teaching protein structure and function. And so what we're going to do is I'm actually going to bring up the web page, and we're going to put Shuchi's going to put the link for the web page that we're using, so that you can actually bring that up. And I'd like you all to do that now. So this is the structure we're looking at. Once again, is as Stephen mentioned, this is PDB entry 6 Lima uniform 7, and the link then should be in the chat at this point. Thanks very much, Shuchi. It's a real privilege and honor to work with the team at the RCSB Protein Data Bank. I've learned a lot and had a lot of fun. So first, we're going to start by looking at the elements on this page. And one of the goals here is that when this is all done, you'll take it back to your campus or to your research group and encourage your students and your colleagues to actually use these resources to tell stories, much like the story that Stephen just told so beautifully. So if we look here on the COVID-19 main protease page for this structure, you'll notice there's a number of tabs on the top. I'm not sure if my mouse is obvious to you, but we have the structure summary, and we'll come back and visit this in quite a bit more detail in just a couple minutes. We also have a tab for the 3D view. And the 3D view actually takes us to a page that includes the MolStar visualization of this structure or of any structure that you're working with. And so that's the second tab. The third tab is annotations. And so the annotations in this particular case, there are 5 links to annotations to different these are integrated with active databases around the globe that provide useful information. Once again, students in any number of different kinds of classes, biochemistry classes, physiology, anatomy classes, pharmacology classes, immunology classes, can use links here to develop stories and to build their stories. And we also have information for experiment here. I know that there are a significant number of biochemistry and biomolecular biology educators who actually are crystallographers. And so they're more likely to use these tools than people like me who simply admire crystallography and use the information it provides. There's also a tab for sequence, and we'll come back and visit this and dig into a little more detail as we get later, but this is interactive and very powerful. There is a genome tab here. In this particular case, we don't have any genomes alignments available for this particular genome yet. And then there's also a version document that tells the history of where, when this was entered, first submitted in 2020, and changes that have been made, adjustments that have been made over time to improve the quality of this PDB entry. And at this point, before we go any further, I'd like to ask Kirsten to bring up poll question 1. And the poll question is, which of the following elements on the COVID-19 main protease structure summary page do you plan to explore further? So we'll take a minute. You can go ahead and answer that and encourage as many of you as possible to go ahead and reply there. And I believe it's set up so you can check more than one box. So please do that. Okay, we'll give people about another 20 seconds to finish their entries here. And Kirsten, if you'd go ahead and end that poll and share the results very quickly. And it's interesting, the most popular options here are the 3D view and the sequence. That's not surprising. And once you've seen, maybe you're familiar with these, but if you're not, once you've seen what the sequence view can do, it's really quite powerful. All right, so I'm going to go back now to the structure summary tab. So please go there, and we refer to this as the SSP, acronym for structure summary page. So if I say SSP, that's what I'm meaning to refer to here. And so I just wanted to go over some of the parts that are on this page. On the upper left-hand corner, we have the, basically this is a link to all of the visualization that's associated with it for the structure itself. But notice we have two options here under assemblies. One is the biological assembly 1, which is shown here. And then there's the asymmetric unit, which is shown here. And so another thing to notice that there are a lot of these question options on the RCSB PDB site. You can click on those questions and they'll take you to other resources that we help you learn more about what's going on there. So if we look here, we have options like we can look at the structure that would take us to the same page as the 3D view tab takes us to. Also the 1D, 3D view, and we'll get to that toward the end of my part of the presentation. Electron density, the validation report, the 3D view, and finding similar assemblies. And as I said, we'll get to those toward the end of my part of the presentation. But I wanted to emphasize a few more features on the page. One of those features is the WWPDB validation here. I still remember when I first did a sabbatical with folks at the RCSB Protein Data Bank back in 2001, I went in thinking that if it's in the PDB, it must be perfect. And I learned very quickly that in fact there's uncertainty in a lot of the measurements in the structures that are determined, and I learned why. And then I asked, well, can we rank them? Can we provide some information? And in fact, over time, that's exactly what's here. So this tells us the parts of the structure that are the most reliable. They show in the blue here, and the parts that we need to be a little more cautious with in interpreting the structure. Also, if I scroll down the page, we also have literature information here. You can link out to other articles by the authors here. These are live links. Go to PubMed and find this. The document object identifier will take you, I believe, to the article itself. And notice here also, there are links to the other PDB structures that are part of the primary citation. And that can be very helpful, particularly if you're looking at a case of a structure that has multiple mutations that make up part of an article. We also have a section here called macromolecules, and the macromolecules include a lot of interesting features here in the macromolecules. I'm an enzymologist, so the enzyme commission classification numbers that are shown here under details are very helpful. Also, we have links out. You can actually go to the UniProt knowledge base for information on this on this protein and its related structures. You have options to looking at sequence clusters by percent identity, which can be very helpful. Also, the sequence reference here, this is a dynamic link or a dynamic object. And notice that when I scroll down on my mouse pad, you can actually see eventually that the residues will appear here on the top. So residue 140 is a cysteine, for example. So dynamic interactive tools you can use. And the last thing I wanted to point to here is the biologically interesting molecules section. And we'll actually come back and visit this in a few minutes. Okay, the next thing I would like to go to is actually the 3D view. So I'm clicking on the 3D view link, and I encourage you to go there now and to manipulate your screen so that the entire MolStar window is present here. And if we look at this structure, you'll notice I'm going to go over here and click on the ligand, which is right over here on the structure. When I do that, it automatically expands to show the ligand itself and its non-covalent interactions with the enzyme. And you can see here that as I mouse over this dotted highlight, this is a hydrogen bond between the ligand PJE5 and a leucine on the structure. And so that kind of information is available. And go ahead and give that a try. Give that a try. And if I want to go back to the original view, I'm just going to reset the zoom here. Takes me back to my original structure. And one of the things that I like to do when I'm looking at structures in MolStar is I go over here to the right-hand side where I can control what's seen and what's not seen. So I like to click on the waters to make those disappear. I know for crystallographers, those waters play very important roles, but for my teaching, sometimes I use the waters, but mostly I won't. Now I'm going to go back to the structure summary page and actually use the links there to take me to specific features that are present on the using MolStar. So if I click on 1D, 3D view, it brings up a window where we have the sequence information on the left-hand side and a MolStar window on the right-hand side. Now notice it doesn't have all of the different tabs and buttons and so forth from the MolStar viewer. But for example, we can go in here and I can click on, notice we have on this screen, we have active site. So I can click on this residue. When I click on this spot, it actually brings in that active site residue, and you can see where that active site cysteine then is covalently attached to the inhibitor. All right. And notice when I'm at this point in the screen where I've got my mouse over that residue, which is a cysteine residue, if I scroll down, it expands the view so that I can actually see that that residue is a cysteine. All right. So the The listing is here on the top, and it expands that view to show that. And if I move my mouse off that cysteine, you can still see that that is highlighted in this tool. And there's a lot of interactivity like this in the structure. If we want to see the other active site residue, we click on this histidine. And here's the histidine that we're looking at, and you can see it has a hydrogen bond to that cysteine. All right, and so that feature is there, and there are other features here. I'm not going to spend time going through all of them, but just point out that you can go to any of these. You can go to one of these bars for PFAM, and it will click and bring up the part that's featured in PFAM. You can look at any of these sections. Anything that highlights like this will bring up an active view in the 1D, 3D viewer. Or we're calling it now the 3D protein feature view. All right, I'm going back now to the structure summary page. And the next thing I'm going to do is click on electron density. I'll give you a moment to find that. So if I click on electron density, it brings up a view of the protein structure. And I'm going to make the water disappear first. And then if I go and I click on any part of the structure, it then reveals the electron density for that part of the structure. And so that can be very revealing when you're explaining this to your students, and they'll notice, oh, wow, this phenylalanine ring doesn't have much electron density associated with it. And so you can use that as a teaching tool. One of my favorite parts right now is the global symmetry link. If I go to that link, so I'm back on the structure summary page. We go to the global symmetry link. I'm going to click on 3D view. And it takes us back to the MolStar viewer. But what we have now is a version of it that includes the symmetry cage. So what I'm going to do now, and what I encourage you to do, is to grab the structure and rotate it toward yourself. There's a rod in the middle of the structure, which is the symmetry axis. And it has an ellipsoid on the end of it. So rotate that to get that in the center of your screen. All right. And so what the software developers at the RCSB PDB have done is they've actually included some keyboard shortcuts to rotate the molecule. So if I press an E, I get a clockwise rotation. And if I press a W, I get a counterclockwise rotation. Now, this is a very handy tool for explaining that symmetry in protein structures is not mirror symmetry, but rotational symmetry in most cases, or translational symmetry. And if you take your students to this page, they can see very clearly that this loop that's extending beyond the cage on the top reflects the loop on the bottom, but it's not a mirror symmetry, it's a rotational symmetry, and they can see that they can rotate those to just switch position. All right, going back now to the structure summary page. And in that same box with all the other links out for MolStar, we also have a link for find similar assemblies. So I'm not sure if I'm going too fast or too slow on this, but hopefully at this point you're back on the structure summary page. And you can click on find similar assemblies. When we do that, it brings up a list of 1,036 assemblies, which are similar to the COVID-19 main protease. But let's say that I want to restrict this search and only look at the ones from the MERS virus. All right, so I come over here to the left-hand side where it says scientific name of source organism. I click the box next to Middle East Respiratory Syndrome related coronavirus. Then I click this play button next to refinements. When I do that, it refines the search. So now I only have 41 assemblies to look through. And then I can look at those ones from the MERS virus, the MERS COVID virus, and explore that. And all the other refinements, if I go back to my previous view, all the other refinements that are present there, you can go down and you can select multiple ones and they'll all be applied at the same time. And that's a very powerful tool for getting groups of structures or for teaching students how to select groups of structures for a report or a presentation. All right, and at this point, I'm going to ask poll question 2. And that question is, which features of Molestar have you explored before? So the poll is open. We'll run this poll for 30 seconds. And once again, you can answer more than one of these boxes. They're all click, they're multiple clicks, multiple options. All right, about another 10 seconds here. All right, I'm going to go ahead and end the poll. Oh, there we go. Share the results. Oh, I just hit stop sharing. Okay, so the results are that most folks have used the structure, but the other options are less frequently used. And I would certainly encourage you to explore those and find which ones reinforce the things you're teaching, or particularly which ones address misconceptions that the students might tend to have. Another feature of the Protein Data Bank that I wanted to emphasize here is the Browse Annotations feature, okay? And the Browse Annotations feature actually takes you to a wealth of connections to all kinds of resources that are out there that we can use. So, for example, ECOD is the evolutionary classification of protein domains. And notice that we have trees here. You can click on these and you can go down the scaffold and look for particular things at as fine a resolution as you would like. The one that I use frequently is enzyme classification. And so we have our 7 EC classes here. So if I go in here, I'll enter trypsin. I'm doing that because I've done it before. Now notice I enter trypsin. I have to click on one of these links. If I just hit the return button, nothing happens. But if I click on this link for trypsin, then it actually expands the tree, and you see you can go now to the entry for the EC class for trypsin itself. And there are other tools here. I know it's not so important now, but back when I was a grad student, mesh headings were important for being able to track down and find literature. Back when we used to look through the Science Citation Index as these great big heavy tomes that were in the medical library. We can look at molecular function. We can look at the symmetry, and I've spent a lot of time there. And you can look here at global symmetry, at localized symmetry, but also at pseudo-symmetry. Some structures that have somewhat, the symmetry is not 100%, but it's close enough that it's worth comparing. And there are lots of resources there for that. So we're going to go to poll question 3. I'm curious about how many people have used annotations. So which annotations do you think would give your students the best opportunities to learn more about protein structure and function? And we're asking you to select up to 3 choices out of I think there are 10 choices for this one. I went a little overboard. So I'll give you another 30 seconds on this one. Okay, 10 more seconds. All right, let's end the poll and share the results. And very interesting that people are interested. Well, enzyme classification and molecular function are obvious, but there's some interest in interest in the other items here. And I'd encourage you to explore those and find resources for your teaching or for learning for your students. All right, now I'm going to go back to the structure summary page. And I think the last thing I'm going to do before I turn things over to Shuchi is look at the ligand interaction. And so what we'll do is we'll go down the structure summary page and get to the biologically interesting molecule interface. And here you see you can actually query for this structure. You can also, here's the full name of the structure. If we click here, we see the stylized representation of this small molecule. We've seen these in, for example, I believe this is the same representation you see with ligplot. And if I click on ligand interaction, that's going to take me back to the MolStar viewer, but with the view particularly for this structure. All right. So what I'm going to do to get a better view of the ligand interactions is first I'm going to hide the backbones, the proteins. So I'm hiding those. I'm clicking on the eye next to cartoon by polymer, and the protein chains disappear. I'm also going to hide the waters. Now I'm going to take my mouse and right click and move things over. Actually, I'm going to rotate the structure a little first. I'm going to right click. So I've got one molecule or one of these in the center of my view. And as I go in here, you can do some interesting things in MolStar here. And for example, if I click on the buttons next to the focus surroundings 5 angstroms, so this brings up all of the parts of the protein within 5 angstroms of the ligand. If I click on those 3 dots, I have an option here. I can hide the ball and stick representation, okay? And then I can just see the non-covalent interactions, or I can hide those interactions. So I have options in terms of the detail. And when teaching this I find this really helpful because it reduces the amount of information on the page. So you can be selective on what you're looking at. But again, these are all interactive. So if I mouse over this hydrogen bond, it shows up on the bottom of this on the screen as a hydrogen bond between a tyrosine and an aspartate residue in the structure. And I think at this point, I'm going to ask one more poll question and then turn things over. Actually, poll question. Okay, as we explored the ligand interaction, which of the following tasks were you able to complete? So go ahead and answer that. I'll give you 30 seconds to answer that question. Super. All right, let's close the poll. And you can see the results there. And it looks like better than half of the people were able to get most of the things to work. The right click and drag, there's some of the other things are a little more challenging. And I'll be honest with you, I teach mostly general chemistry these days. And so I actually went in and spent a fair amount of time practicing this to make sure that I could get it right for the presentation. But I'd encourage you to go in and practice that. So let's close that poll window. And one more poll is about the 1D, 3D view, okay? So question is, for the 1D, 3D view screen we looked at a couple minutes ago, what features of that screen are you most likely to use in teaching? And once again, we'll have 30 seconds to answer this. All right, and we can see from the results that the active site is the one that has the most people interested in using, but secondary structure is also there. And I would encourage you to spend some time on that screen to become more familiar with it, so that you can use it and teach your students to use it. There's a lot of useful and dynamic information in there. At this point, I'm going to turn the presentation over to Shuchismita Dutta. I had the privilege of working closely with Shuchi for about 4.5 months in the spring of 2022. I really enjoyed that and learned a lot about her and learned a lot about appreciating her efforts as an educator and her focus as an educator on really helping the faculty learn how to teach our students. So Shuchi, I'm going to stop sharing.
Thank you, Paul. I am happy to, take this webinar forward, but I see there's a lot of question answer happening in the chat. And I wanted to pause here and let people ask. So there are a couple of questions about saving images. There are a couple of questions about I think there was a question where somebody, I think, was a little lost because we went too fast. So before I go to the next part, I wanted people to give a chance to ask the question again. Paul, did you want to share something about saving images? Or do you want me to do that?
Why don't you go ahead and do that, Shuchi?
Okay. So I am going to share my screen. And so Paul was sharing this particular PDB structure, 6LU7, and we were looking at the structure. As soon as it loads, okay. And he was, clicking on the ligand to show various interactions. So in here, if you look at this particular lens icon, the screenshot, this is what will allow you to download an image. So you can capture whatever frame you think is suitable for your particular interest. In addition to capturing a still, you can also capture animations. And so if you click on here at the bottom on the right-hand menu, you can define the animation trajectory, whether it's a loop or it's a rocking. You can specify how long you want it to be and save an animation. So that is also possible. There was a question about selectively showing specific types of non-covalent interactions. That was also, I think, a very important question, particularly in a teaching context. When your screen is busy with all these different kinds of interactions, it's difficult to discern which one is hydrogen bond, which is salt bridge, etc. So what you can do is that if you expand this option set here and look at non-covalent interactions, it'll show you all of these. Right now, the ionic interactions are actually turned off. So notice when I turned it on, this shows up. And I can selectively turn on and off any of these to only show the type of interaction that I am interested in, or I want to discuss, or I want my students to focus on. So there are all these options that are also available that allows you to sort of make the most of this display and discuss the aspects that are relevant to your particular topic. Okay, are there any other questions? I'm sorry, I closed the chat. Okay, yes, and I see that Orla is saying in the chat that PDB 101 has several tutorials, and there is actually also extensive documentation. So this little help button, if you click on this, it'll actually take you to documentation that gives you all kinds of scenarios and detailed instructions for, and so the key controls that Paul was using earlier, they are all explained here for Z and moving and rotating and all these things. These are all shown here. There is an extensive FAQs, scenarios, questions list that I encourage you to scroll through. How do I color? How do I change this? How do I do this? So feel free to go through all that. And if you still have questions, don't hesitate to reach out to us. We would love to hear from you, and we would love to, if there is a question that you don't see answered here, let us know, and we will be happy to add those things. Okay.
I'd also like to insert at this point, Shuchi, I'm going to be at Discover BMB in San Antonio, and I would love to meet and talk with any of you who are there. So please reach out to me, and I'd be happy to engage in San Antonio over breakfast, dinner, whatever.
Great, thank you, Paul. So I'm going to continue the rest of the webinar as planned. And in this section, what I'm going to do is do something So Stephen introduced the topic, the idea. Paul showed you the various features, functions, tools that we have. I'll show you some more tools. But we have all these molecules, and they are doing very incredibly complex activities, functions. They are interacting with each other, performing specific actions, whether it is catalysis or recognition or whatnot. And what I will do here is hopefully, sorry. So what I will do here is in the form of a molecular story, try to engage with you. And so my thinking is that when you're teaching, one very important component of getting your students to pay attention is to hook them or hook their interest with some kind of a story, something that they care about, something that they want to know or learn. It's only then that they are likely to pay attention to what is going on. And if you can encourage them to ask the questions and be curious, that's really the place where learning happens. And hopefully that's also the place where they will remember what they did or will be able to find their way back again. Instead of just doing a worksheet where they say, click here, do this, save this, open this, et cetera, et cetera. So founded on this motivation, I lead this molecular CaseNet project, where the goal is to both develop and use these molecular case studies. And the idea is that you are looking at a real world problem at the interface of biology and chemistry. And I want to give a shout out to Henry. I don't know if he's still here. Henry was the person who guided me to this interface of biology and chemistry idea. And the whole operation around this cycle that I'm showing here is that, let's say you come in with a specific question in biology. This is your research question. And this is set in a particular context. So today's context is going to be pandemic to Paxlovid. And that's what we will talk about in a moment again. But once you've asked the question, you have to identify the molecules, complexes, whatever is relevant to that particular topic, and then use the data, tools, and resources that we are learning about to try and answer this question. And so what you're doing is going through this cycle at least once, and that is what is a required element of a molecular case study. We call this the molecular case study cycle. And so in order to set the context and ask this question in biology, I'm going to take you to the next slide. But before that, I wanted to sort of remind you a little bit about the biology of these coronaviruses. The coronaviruses, as Stephen mentioned, so they have these spike proteins that bind to the ACE2 receptors. That is internalized, and then the genomic material, the positive stranded RNA, is released in the cell. Now, this positive stranded RNA is essentially an mRNA that will get translated, and it will produce the proteins, the viral proteins. However, the catch is that this protein that is produced is a long polyprotein. And so all these key enzymes that the virus needs to replicate itself and to carry on the rest of the life cycle, they are produced in this long polyprotein, which is non-functional until this polyprotein is cleaved into these little pieces that are the papain-like protease, the main protease. Here we have the RNA-dependent RNA polymerase. So the rest of the life cycle will not happen unless this polyprotein is chopped up into these little pieces. And that is where the role of the main protease is. This is what Stephen introduced earlier today. And so this inverted triangle is very key. And if we can somehow inactivate it or make it non-functional, then the rest of the life cycle will not happen. And this is indeed the heart of what the Paxlovid drug is doing, the active ingredient being nirmatrelvir. And so the reason why this is also a very good target is because notice how this particular molecule has to act on these 10, 11 different places in order to make the whole life cycle proceed by liberating all these different pieces of the genome. And not only is the enzyme active site highly conserved, these points at which the enzyme acts, they are also highly conserved, because if either of them change, the viability of that virus reduces. So that makes it a very good target. And the other interesting thing that is worth noting is that in many of the coronaviruses, the SARS-CoV-1, the MERS, and the SARS-CoV-2, the protease, this main protease is highly conserved, as is the sequence at these particular sites. So if you think about it, if we could design an inhibitor that works to inhibit this protease, perhaps it could help us get over not only SARS-CoV-2, SARS-CoV-1, MERS, and perhaps other coronaviral infections. So with this background, I'm going to go forward and take you to this picture. So this picture is the picture of the Pfizer scientists, the team of Pfizer scientists who developed the drug Paxlovid. And this team is led by Dr. David Owen. And the case study is designed based on an interview that he recorded with the Forbes magazine. And in this magazine, he talks about how in mid-March, when the whole world went into lockdown, the CEO of Pfizer called him up and said, what would you need to create an oral protease inhibitor that can be made available in the market? So let's imagine you are part of this team, okay? And let's look at a little bit more of what he said in that interview. So this whole interview is, of course, available with the case study if you were to do the whole thing. But I want to draw your attention to this particular statement that he makes, where he says that the SARS-CoV-2 main protease catalytic site, when compared to the SARS main protease that appeared 17 years earlier, is pretty much identical. And so this 835231, this particular molecule that they had designed 17 years ago, he thought that this is a great starting point for designing an oral protease. The catch here is that this drug had gone through initial screenings, had not gone through the phase 2, phase 3 clinical trials, because there was no pandemic at that time. It was an epidemic which was contained, and it was gone by the time they had the drug ready. So they had no patients to test it on. And number two, this was developed at that time as an intravenous drug. So this was not an oral inhibitor. And this is also an opportunity to talk about how designing a drug to be an IV administrable and an oral administrable drug, that there are different ball games, different things have to be considered and understood. So what I'm going to do over here is, as let's say we are part of this team, can we, with the data available in the PDB, can we justify this idea that this drug, this 835231 drug, is a good starting point for designing an oral protease inhibitor? This is what we are going to try and examine. And before I go any further, any questions? Okay. So what I will also do is I'm going to share. So how many of you have not seen the document? Sorry. So I'm going to post this in the chat one more time. So if you want to follow along, I think the steps are listed over there. But I'm going to go back and share my screen. Okay, so if you did not find the document, please type in the chat and I can post it again. But what I'm going to do is we're going to say, like, I know nothing about this particular molecule. Okay, I don't know what it looks like. I don't know whether it is even in the PDB. So what if I search for this and see what I can find? The couple of things that I would want to ask is, like, if this molecule is available in the PDB, does it bind to the SARS-CoV-2 main protease? Before I use this drug as the foundation for designing new features and functions, the first order of business is, does it even bind to SARS-CoV-2? And what else does it bind to? Does it bind to other things? Because if I'm going to put in a whole lot of effort to design an oral inhibitor drug, I would want it to be able to inhibit not just SARS-CoV-2, but perhaps a few other coronaviruses, because we don't know how this virus is going to continue mutating and changing. And so with this idea, I'm going to open a PDB web page. And let's see how this goes. So I'm going to go to the beginning of the thing, and let's say there are so many screens and buttons and things going on. Sorry. So this is the drug. So I'm going to just copy the name, and you have that same name that is available in the document that I shared. So I'm going to just put that name in the search box at the top of the page on the PDB website. And you can already see that there are things that are showing up. So likelihood is that there are some structures in the PDB with this molecule. So let's just run the search and see what I can get. Because right now I still don't know what is the ligand identifier. What is this molecule? What does it look like? And so once I run it, I see that there is this porcine delta coronavirus with the inhibitor that I'm searching for. So let's open this up for now, because what I want to do here is I want to identify what is this molecule. So learning from what Paul showed us, we can scroll down on the structure summary page to the small molecules section, and notice there is glycerol, there is DMSO, and then this is this molecule, which is that particular Pfizer drug. So let's open this up. I'm going to click on this V2M molecule. And this is the ligand summary page that shows you what this molecule looks like, the chemistry of it. This is an interactive 3D molecule that I can rotate around and see what this molecule looks like. And also here is a listing where if I click on this search, any instance, any structure in the PDB that has this particular molecule, this drug, this inhibitor present in the structure, I can look it up right by clicking on that search button. You can look through the rest of the page. It gives you all kinds of identifiers. And if there were links to other resources like the ATC or Drug Bank, etc., KEBI, things like that, it would show up here. But this is not an FDA-approved drug, so we're not going to find much here, except what I am interested right now is the question that, does this molecule bind SARS-CoV-2? This is the question I'm asking, and let's see what I find. So when I run that search, I'm going to look at the column on the left and notice that there are 4 structures in all, 2 of which have are SARS-CoV-2 proteins. One is that porcine delta coronavirus, and the third type is the SARS-CoV-1. And so now, what do you think? Do you think that they made a good judgment in saying that this is a good starting point for developing or designing a new drug? What would you think? Yes, no? So I see a lot of yeses popping up. And so by just finding a structure with this particular drug bound to it doesn't necessarily mean that, we've gotten it all. Because we have these X-ray structures, I think it's worth looking into these structures to ask 2 questions. One is, where is the drug binding? Is it really binding at the active site, or is it binding at some unrelated, remote site? Because that will not be helpful. And 2 is, can we actually convince ourselves that indeed these target proteins are really similar or different. So let's approach this one at a time. So here I'm seeing this particular drug molecule bound to SARS-CoV-1, and here it is bound to SARS-CoV-2. I'm going to open up both of these structures in different tabs. Okay. So let's look at this one. This is SARS-CoV-1. And what I'm going to do is I'm going to scroll down to the small molecules section, like Paul showed earlier, and click on that ligand interaction. So this is going to show how this drug molecule, this is the drug molecule, and how it interacts with the enzyme. Just for the sake of making it easy to look at, I'm going to select this and color all the carbon atoms a different color. So now our ligand is going to stand out against the rest of the protein, and I can see that here is my catalytic cysteine, the residue 145, and there is the catalytic histidine. So this is the catalytic dyad. This inhibitor is covalently bound to that catalytic cysteine. And so that's what is happening in SARS-CoV-1. This is the SARS-CoV-1 structure. Let's look at what's happening in the SARS-CoV-2. We can repeat the same steps. And I hope that is helping you get familiar with this. And so again, here is our drug. I'm going to again color this in a different color. And so I'm going to select all the carbons from the drug and color it again magenta. And again, we can see the covalent cysteine binding, and that's our histidine forming the catalytic dyad, right? So here's the cysteine, and that's the histidine. So the interactions are very similar. So that's encouraging. It's encouraging that the drug is, first of all, binding to the active site, and it's binding in the same manner in both SARS-CoV-1 and SARS-CoV-2. Now, you may have heard about scenarios where the enzyme catalytic site is conserved, but the rest of the enzyme is different. And we had seen in that interview, Dr. Owen had said that the structures are similar. So can we convince ourselves of that? And to do that, so I see that Jay is asking, is there a structure with a natural ligand or a substrate analog? So the structure that Paul was showing is actually a substrate analog. So that 6LU7, you can compare it with that, and that does bind the same pocket. It does make the covalent linkage with that cysteine 145, as Paul was showing. I hope that answers your question, Jay. Now back to our homepage, and what I'm going to do is I'm going to open this pairwise alignment tool. And I'm going to compare 6XHL and XHM using this tool. So again, if you're on the home page, you can go to Analyze Pairwise Alignment Tool. And this opens an interface where I can put in just two PDB entries, XHL, 6HXM. And I'm just going to use the chain A default settings and run the comparison. And voila, this is what the chains look like. Sorry, this is something else. XHL. So I put in the wrong. Apologies. XHL and XHM. I was wondering why the protein looks so different. Yeah. So here are the two chains, one orange and one blue. superimposed on each other. And here's the sequence. Again, this is interactive. I can zoom in and see what is going on. A summary table above says that the root mean square deviation is 0.36. This is 1/3 of an angstrom different, the backbones. So it's pretty much identical. So there's really nothing different in these two. And I can bring up the other chain in the dimer. The protein functions as is active as a dimer. And if I show the ligands, they all pretty much superimpose on each other. So indeed, they were right in their assessment that these two proteins are very similar. Their active sites are identical, and this inhibitor binds to both the enzymes equally effectively. And so that's a great starting point for them to go ahead and design their drug. I'm going to again pause here and see if there are any questions. So how do I get to the structure comparison? If you go to the Analyze menu up on top, in the top bar, and click on pairwise structure alignment. That's what I'm using over here. Okay. Any other questions? Yes, indeed. This is a very, very tool, and I'm a big fan of this thing. This allows us to do lots of interesting things. Now, what I want to do in the next part is to take you through and ask one more level of questions. And so I showed you how we compared the different proteases, the active site and the overall structure. And I wanted to summarize here or review here that we used the top search bar to find the drug. We identified the different enzymes that binds this particular drug. We also examined the nature of the inhibitor binding, the kinds of interactions it participates in binding to the drug. And so these are some of the skills that we learned in doing all this in the context of this interview that we are trying to justify or understand. Okay. So the next part actually takes you to the rest of the interview, somewhere in which they're talking about how the preclinical team got a sense of this. And so by now they have developed this new molecule called nirmatrelvir. So this is a different number, 7321332. This is what is the active ingredient in Paxlovid. And they have established oral bioavailability, done animal model, safety margins, etc. So now the question is that if this were to go into the market, does this bind to the various mutants of SARS-CoV-2 that have been floating around? And what is the fate going to be if this coronavirus actually mutates enough to become a new, different coronavirus? Would it still work? So these are, I think, very valid questions that you would want to ask before you can accept this in the market as a viable drug. And so this is, again, what we could do. We could go into the PDB and, again, use this, much like what we did for the previous round, use that drug name to search and see what we find. And this time, the ligand ID is 4WI. So I'm going to just jump into 4WI. And I wanted to show you a few features and functions that are beyond examining one particular structure. And these are looking at groups of structures so that you can look at trends and patterns and hopefully make predictions and or develop hypotheses that can become the basis for new questions that you can ask. So again, in the same manner that I had shown before, I'm going to run this search and find a whole bunch of structures. So there are, with the nirmatrelvir bound, there are 34 structures in the PDB. Now, notice, 34 structures are already too many. I'm seeing that it is, binding to the SARS-CoV-2, but also MERS, also SARS-CoV-1, and this 229E, another coronavirus. So that's all good news that it is binding a variety of different coronaviral proteases. But as I scroll through here, I will see that there are examples of Omicron with this particular mutation, lambda with that particular mutation. And so there are a variety of mutants that are also binding nirmatrelvir. And how do I make sense of all this data? There is just 34 structures is too many. So how about we group these structures by sequence? And for simplicity's sake, I'm going to group them by UniProt ID. So as we did the search, this search box got populated, and it's saying that, it's looking for structures that has 4WI, either covalently or non-covalently bound. And now I'm saying, show me the results, not as a single entry at a time, but a group, and show these groups. So instead of seeing 34 structures, I'm seeing 5 groups, okay? And this is the largest group. It has 21 structures. The rest have 1, 10 other structures. So I'm going to open up this group. Now, this group, if you remember, is by UniProt ID. This is going back way at the beginning of the talk. I had shown you that it made this long polyprotein. And this is actually the UniProt matching that long polyprotein. So looking at this group, there are 21 members present in this group that match our search criteria. You can figure out that there are, these are all X-ray structures, and when were they published, what is the resolution, et cetera, et cetera. But what I am more interested in is actually looking at the group sequences. And what I see is this. So all the structures that matched my query are listed here. So if I may have the last polling question, the question is that here we have the whole UniProt sequence spanning across the whole entire box, and our matched sequence is just a small part of it. So why do you think that the polymer sequence lines up only this small part of the UniProt? Right, I think, okay. So I'm going to stop the poll here. Thank you. And the answer is that indeed that the complete UniProt sequence actually has all the pol proteins. We are only looking at the protease, which is just this much, okay? And so that's why all this is matching. And just like Paul showed earlier, if you zoom in, you can actually see the sequence show up over here. Zoom in a little bit more. But notice this particular residue, for example, if I click on it, this is that active site cysteine. So there are structures which have mutations there, shown by this pink bubble, and there are others that do not have the mutation. But bottom line is that right now we are looking at a group of structures that have nirmatrelvir bound that are in this group. And what I do want to show is what we were looking at was this 1D alignment, but in the same spirit as we had the 1D, 3D alignment, we can look at this group in 1D, 3D. And I think this is really powerful because we can now superimpose any of these by just clicking on. And it shows up over here. And I can say, oh, I want to, zoom in on this residue. And it'll focus in and show you what is going on over there. So of interest is this catalytic site residue. And you can ask questions like, what would happen if that cysteine were mutated to serine or some other thing? Would the drug bind? Would it not bind? What can you learn from that? And towards the end in your, in your script, as you go along, there is a question asked. So there is a challenge question asked at the end, is that, can you identify any SARS-CoV-2 main protease mutation where this nirmatrelvir drug would not bind, right? And the answer to that, I think, can be gotten to from this kind of an analysis where I'm looking at the active site and anything close to the active site, right? Yeah. So for example, this particular residue, which is just 2 residues away from this catalytic cysteine, I'm going to open this up, okay? And so 8E26, I'm going to open it up in a different window. 8E26. And this particular mutant, notice that in the title of the article, they're already looking at structures bound to a different inhibitor. And in fact, this is an alternative to nirmatrelvir that is coming up that a Japanese company has developed and is marketing, and it's on a fast track in FDA approval. So the idea that these protease inhibitors can be designed and can be put into the market, tested and put into the market, but then once with use, these drugs become, ineffective because the bacteria acquire mutations that bypass this particular drug. Newer drugs may have to come in. And so this idea can be explored and discussed in a manner just by looking at the data, just by understanding the active site chemistry, the active site, like the shape and physical, interactions between the drug and the inhibitor. So all that I feel is enabled using the data tools and resources available from the PDB. And I encourage you to try this. And what we have done here is on top of the skills that we talked about in the previous research question, I introduced you to the idea of grouping structures. Now, grouping structures gives you the power to say, not just what one structure is doing, but the ensemble. What is it telling you about the biology? And can you use that knowledge to either ask new questions or explain other types of data that you have gathered from other resources or from experiments, etc. And with this, I'm going to draw you back to this large array of proteins, all of whose stories are waiting to be told. So if you are interested, we would love for you to participate in writing up or creating more stories that can be brought to your students, hopefully engaging them and giving them a real life context for why they need to understand the enzymology, what is the critical deal about thinking through this specific interactions and designing inhibitors, etc. I'm going to pause here and see if there are questions. I know, Paul, you've been answering questions. So Seema asks, how could I go to the group sequence? So there are multiple ways in which you can do that, Seema. So what I did in this particular case is I actually ran the search. Let me see if I can do that. So I was running a search, and in the return answers or return results, I changed the response from structures to polymer entities and selected groups. Another way in which you can look at groups is, if you remember the 6LU7, the PDB entry that Paul was showing you. Here, if you scroll down to the macromolecule section, here too, if you click on this particular, I'm not going to do it because it's going to pull up 3,000 structures and that's going to, block everything. But here's another place where if you click on this or this UniProt group, so these are groups by sequence identity. This is grouping by UniProt. So if you click on these, it'll give you that listing of grouped sequences. I hope that answers your question. And any other questions? Feel free to unmute and ask. Okay, if not, I'm going to try and wrap up this presentation. And say that today, Dr. Burley introduced you to this idea of how data from the PDB was relevant and useful in both designing drugs like nirmatrelvir and also in the design of the vaccines. We then got a tour of the various features, functions, and tools that are available for you to explore. So Paul gave that tour of 6LU7, your favorite structure, your favorite molecule. I would encourage you to play with the structure summary page and the various other tools and functions we introduced to you. And hopefully this molecular storytelling session from pandemic to Paxlovid actually helped give you ideas of how you can engage your students in interactively using these tools to really ask meaningful questions and answer them and find answers. We have collected a wide variety of information from many different bioinformatics data resources that we connect to and we make available from our website. So feel free to explore those. And if you run into trouble, if you have questions, if you have ideas, don't hesitate to reach out to us.
I have a question.
Yes, go ahead, Arpita.
Yes. So my question is that, I think I missed that part. This is so interesting, but, when you typed the name of the small molecule inhibitor that Pfizer had, I missed that part. How did you get to the unique protein name, for example, 4WI? I was trying to do that, but I missed that portion.
So the molecule, I'll just do that quickly so that you don't feel lost at the end of this session.
Yeah.
Here, if you just take this drug name and put it in the search box, and don't worry about anything, just go ahead and search it. So here we are seeing this is the MERS protease bound to that drug. And the only ligand in this particular structure is 4WI. That's how I found it.
Got it. Okay. Thank you. Yes, I just missed that portion. Thank you so much.
You're welcome. So what I have done is I have posted in this chat, okay, not the drug name, but this link for you to complete a survey. We would really love to hear from you. What you thought of this session, where you think you would want to learn more. And I'm going to put it in the chat one more time. And perhaps this can be shared again, Kristen, along with a follow-up email from ASBMB. That would be awesome. And then, of course, we cannot end without thanking the entire team of PDB that is doing very important things and keeping all this going. And also Paul for collaborating with us and helping us put together this session. Thank you, ASBMB, for hosting us. Thank you for advertising and managing all the logistics. So we really appreciate your role and thank you all for your attention. I will close with saying that there are opportunities for your students to apply and in various positions and capacities. So on the PDB website, if you look up jobs, all these details are available. So please encourage your students to apply if they are interested in even like summer research or gap year interns, postdoctoral fellows if you have graduate students. And finally, the recording from today's session will be made available on PDB 101 after we receive it and put it together. But we definitely want to hear your feedback. So I would really encourage you to complete that feedback, that survey, and give back. Paul, do you want to say anything to close?
Just to encourage, stay connected. I mean, if you've learned some things today that you think were really useful or helpful, or you want to dig a little deeper, then reach out. And as I said, I'll be at the meeting in San Antonio, but you can also reach us through the PDB website, or I'll actually put my email in the chat link. And you can certainly contact me. We're always looking for better ways to serve the education community. And we also find that we get extremely valuable input from the education community in this process to tell us what you're using and what you find easy to use or difficult to use, or things you'd like to see that aren't there yet. That's all really valuable input.
With that, I would also like to, invite you to the possibility of collaborating with you. So if this storytelling idea inspired you, and if you feel that you want to write or create more stories about your favorite molecule, we would love for you to work on such a lesson or curriculum, depending on what your interests are. And we would be happy to work with you to bring it up to a point that it can be shared on PDB 101 for other educators to use and learn. And this could become the foundation for collaborating on a manuscript together. So there are all kinds of possibilities. So feel free to consider this. And if you have particular topics, molecules, or stories you want to tell, feel free to reach out. Any other questions, comments?
Well, many thanks to all the people that stayed with us for the whole time.
Yes, thank you so much. And Kirsten, do you want to say anything to close?
I will, but I think Jim has a question that he's been wanting to ask.
Go ahead.
So, yes, sorry. I don't want to keep people here. It's been very interesting, but mention was made early on about, this could be used to help students with misconceptions. So I feel like a lot of what I do in the classroom is to simplify things that are complex, So in this case, we're showing them a lot. And so I'm wondered if there's information about, in particular, what are things that students have misconceptions about that a dynamic structure or, this kind of level of structural information can help them with?
Paul, do you want to respond to that?
I guess I will say we don't have anything ready to come out of the box at this point. Do you have some thought? I mean, that's something that would certainly be worth pursuing, and the whole question of what can we at the PDB do to help clear up misconceptions. I think molecule of the month, if you can find the molecule you're studying in molecule of the month, there's often a nicely simplified and clarified versions there. That's a place I would start.
That's definitely a great place to start. And so if the misconception is that the protein has a particular shape and it is sort of static and this is how it is, I would encourage you to guide your students through the number of structures which have different conformational states. That's one misconception that can be addressed. Then there are misconceptions from the space of biomole business interest, for example, representation of different structural formats. So when you're looking at a molecule, sometimes it appears hollow or spread out, but if you change the representation to surface or space fill, you might appreciate how tightly packed or dense or close together those interfaces are, or the ligand binding is. Or for example, in drug binding, people think that, the drug is just hanging out there with these 3 hydrogen bonds. But often what is not obvious is that there's probably a hydrophobic pocket where the drug is like really snugly fitting. And there are many other interactions that are, supporting or keeping it there. And if you change any of those interactions, that drug will either not bind or not even enter, or other things could happen. And so there are, I think, opportunities with real data to open their minds to all kinds of possibilities and questions so that they can sort of take the ownership of looking at and finding out more instead of them being fed all the answers. You know what I mean? Because at the end of the course, I think all of us want our students to leave the course empowered to apply these tools to any other topic, not just do it for, your Biochem 101 and you just did these 3 proteins and that's that. You want them to use it for any other course that they take, any other project. I hope that addressed some of your.
Yeah, thanks. That's helpful. So immediately, like one little thing that comes to mind is, for example, are there links, or could there be links to a structure bound to a ligand? And then if there is published information about the dissociation constant, or Ki or something like that, if that was linked to that somehow, it would make it a lot easier to find as an instructor rather than trying to look through literature.
So we do connect to binding DB and binding mode. So if there is availability of that information in the section where you're looking at in the small molecule section on the structure summary page, below that ligand, there will be information on links going out to binding DB that you can click on and go directly, and then look through their papers and analysis and discuss in class what is the significance of this. Yeah, that is available. And I see Emmett has a hand up, and I don't know if we are going to allow that. Go ahead, Emmett.
Okay, thanks. I was just thinking about that idea of the complexity of the PDB showing to, for example, undergraduates, which is my, teaching area of focus, and how I think there is some potential, like, especially when we were looking at, okay, you could hide these certain types of bonds or show these other types of bonds. There are like bonds in that list that I don't know what they are. And so I think that's like one of those examples of, I think we got to be, I don't know, I'm just thinking about that particular thing, but I'm sure there are others, this is such a tool with depth, like, because it needs to be. And so, yeah, balancing those things might be a little complicated. I just wanted to point that out as like one particular area I was thinking of.
So thank you, Emmett, for bringing that up. So this has been feedback that I've received from many other educators. And in response to that, some of those interactions, like I, for example, once I started playing with the MoldStar tool, I had no idea what halogen bonds are. And so I'm like, what in the world is that? So I educated myself and I created a video. So now there is a video out there that can tell you what is a halogen bond. And it not only tells you what is a halogen bond, it'll tell you how to identify it in the PDB. And as a result of these conversations, we actually created a collection of these resources. So several educators pitched in. And just last month, we published this box of lessons, which actually takes you through different types of biological macromolecules and tells you what to look for, how is that relevant in biology, how does that show up in structures in the PDB, what can you do by looking at them and learning from them. So it covers proteins, nucleic acids, lipids, and carbohydrates, and also simple things like how to find structures, what are the, going from atoms to molecules to polymers to larger assemblies. So all these things are kind of discussed there. Feel free to check it out. I've posted the link in the chat. If you have suggestions for other types of materials you think would be useful for your class or your students, keep telling us. Input from you is really, really valuable, because we can be sitting up and imagining things that, oh, this is what educators probably need, but really the answer comes from you. So please tell us, what is it that you need? And we look forward to that feedback. Yeah.
Thank you for staying a few extra minutes to answer some of these questions. Thank you, Shuchi and Paul and Stephen, for the very helpful session today. And thank you all for sticking around for a few extra minutes. We've posted the evaluation link in the chat a few times. You'll see that in a follow-up email. The recording will be posted both to the ASBNB website and to the PDB website, hopefully here in the next several days. And in the meantime, if you are using or have developed a similar sort of resource that you think would be beneficial to the education community at large, the education department at ASBNB is always happy to work with you on how we can share this information. So please feel free to reach out to education@asbnb.org. And we hope to see you at a virtual event sometime here in the near future. Until then, have a wonderful afternoon, everyone.
Thank you all.
Bye-bye.
Bye.